Bueno, bienvenidos al segundo día de nuestra reunión de ECOFLOR. Veo que hay, no sé si son afectados de anoche o simplemente que tenían ganas de dormir un poco más. Ellos se lo pierden. Porque hoy rompe el hielo o empieza dando una charla plenaria Timo Niet, a quien muchos de vosotros, bueno, algunos de vosotros ya conocíais, otros lo habéis conocido ahora. Timo es holandés, como habréis deducido de su apellido, pero él hizo su tesis doctoral en Zúrich, bajo la supervisión de Peter Linder, y después ha tenido algunos fotos por ahí. Uno lo tuvo con Steve Johnson, estaba en Sudáfrica. De hecho ahí fue donde lo conocí yo, en el Cabo, en la Universidad de la Ciudad del Cabo, y tiene mucho interés en evolución de plantas y sobre todo en el papel que los polinizadores ejercen en la diversificación de las angiospermas. Y sobre eso es lo que va a hablarnos hoy. Así que nada, Martino.
Buenos días, yo no hablo español, así que voy a presentar mi trabajo en inglés. Good morning, everyone. First of all, thanks very much, Fernanda, for inviting me. I'm really excited to be here, and I really was impressed with the level of the talks yesterday, and just trying to imagine whether a meeting like this would stand a chance in Holland, and we would certainly not be able to fill 2 days with great talks about this particular subject in our country. So I think you're doing fantastic work. Just to explain where I work, because I think most of you are not familiar with Naturalis Biodiversity Center. It's essentially the Natural History Museum of the Netherlands, which contains a large herbarium collections of 3 universities and also some zoological collections that have all been amalgamated. Of course, one of the most striking aspects of angiosperm diversity is the extreme variation that we observe in flowers. And it's really this subject that brings all of us together, especially in our aim to understand how this diversity evolved. Plants have a great challenge. If you look at the distribution of individuals in this meadow, you see that they're sessile, and if these plants want to reproduce through outcrossing, they need a particular vector to transfer their pollen from one individual to another. And about 87.5% of plants depend on biotic pollen vectors or pollinators for this process. And the idea that plants rely on pollinators has been important in creating ideas about how these pollinators may have been involved in the diversification of angiosperms, ideas that of course have been around for centuries. I'm going to start this talk with quite a broad general introduction, providing a theoretical framework of how pollinators might be involved in driving angiosperm diversification. And this theoretical model, which we have, was largely initiated in the 60s and 70s by work by the Grants, and then further helped by work by Stebbins, and then Steve Johnson developed this into kind of a formal theoretical model or conceptual model that is now known as the Grant-Stebbins model. One of the pillars of the Grant-Stebbins model is the realization that not all pollinators are the same. And what I mean with that is that pollinator species vary in several respects. For instance, pollinator species vary in their preference for particular colors or particular scents, and of course color and scent are important traits that flowers use to attract pollinators. So I've just given some examples of this preference here. This is a recently published study on long-tongued flies from the South African flora. You'll see that many of my examples are drawn from the South African flora simply because that's what I know best. But I'm sure that similar examples exist worldwide. And these flies have a strong preference for pink and blue colors of model flowers. But in contrast to that, other pollinator species have preferences for different colors. And the same applies for variation or preference in scent. So this is a study by Rob Pico and colleagues who've done a lot of work on sexual deception in Australian orchids. And this species of wasp has a strong preference for a chemical synthetic blend called Phylo-1, whereas other species of wasps have strong preferences for slightly different chemical structures. And in these cases, these structures conform to the sex pheromones that are produced by the female wasps of their own species. And obviously the male wasps have a strong preference for the pheromone produced by females of their own species. And these orchids have mimicked these sex pheromones, and slight variation in the chemical structure of these pheromones leads to different preferences by different wasp species. Color and scent are traits that plants use to attract pollinators, but there's also variation in other aspects of pollinators that is potentially relevant to angiosperm diversification. And that is that not all pollinators are the same in their own morphology. So this is an example from two species of sunbirds that occur in the Cape Floristic region in South Africa. And I've just lined up the beaks of these sunbirds, and you can see that the one sunbird has a longer beak than the other. And this, of course, is something that's extremely common where pollinators vary in the length of their mouth parts. And this creates a potentially important selective environment in which plants operate. So, if we put the variation in preference and morphology of pollinators in the context of plant evolution, we can come to an important conclusion. First of all, it's important to note, of course, that plants rely on these pollinators for reproduction. So, given that these pollinators differ in their preference and their own morphology, this creates a very strong potential for natural selection by pollinators on floral traits. So, if flowers vary in traits that attract pollinators like color or scent, or if they vary in shape, there's very strong potential for natural selection to operate on that variation. One of the expectations that follows from this is that plants that are pollinated by the same pollinator or that belong to the same pollination guild are expected to express the traits which reflect the preference of these pollinators, or that reflect the shape of these pollinators. So here are two quite stunning examples in terms of flower color. On the left-hand side, you see members of a pollination guild that are pollinated by mountain pride butterflies in South Africa. And you see that all these flowers are extremely similar in their flower color, not only to our eyes, but actually also in terms of the spectral reflectance of the floral parts. And more interestingly, the close relatives of these plant species that are not pollinated by mountain pride butterflies have often different colors. So this suggests that a shift towards pollination by mountain pride butterflies coincided with evolution of red flower color, and that suggests that these butterflies have a strong preference for this color. And the same can be concluded for this pollination guild, also from South Africa, a species of oil-collecting bee. And all the plants that it pollinates are characterized by these greenish, yellowish flowers, and the close relatives of these species, which are not pollinated by this species, are not characterized by this color. So again, from that we can infer that this pollinator species probably has a preference for this particular flower color, and that convergent evolution onto this preference has occurred in these flowers. Something similar has been studied in terms of floral scent. So most plant species that are pollinated by carrion flies are characterized by sulfur compounds. These compounds are the degradation products of proteins, and obviously carrion flies strongly rely on degraded protein or carcasses for their own life cycle. And these plants exploit these pollinators by mimicking these sulfur compounds and deceiving the fly to think that it's an actual carcass. But most interestingly, what you see if you analyze the floral scent of these species and their close relatives is that the species that are pollinated by carrion flies are characterized by sulfur compounds, and the close relatives that have different pollinators are not. So again, we see converging evolution driven by preference of a particular pollinator for a floral attractant trait. And of course, the same applies to floral shape elements. So here the floral tube lengths of flowers pollinated by long-billed sunbirds is given, and these tubes are quite long in comparison to species pollinated by short-billed sunbirds. And again, these are often fairly unrelated plant species that have all converged on the beak length of the same pollinator. So I hope I've convinced you that pollinator preference and pollinator morphology play an important role in selecting for particular floral traits. But the model for pollinator-driven diversification has a second important element, and that is that not all pollinators are evenly distributed throughout the landscape. And of course, this is obvious if we think about the distribution of hummingbirds, for instance, which are restricted to the New World, and some birds which are restricted to the Old World. That's on continental scales, but even much more local scales. If we travel through the landscape, we often observe a turnover in pollinator species, not only at the species level, but perhaps sometimes also on the level of functional groups. And this is just an example from a study that, of which many are available, showing that pollinator communities vary spatially, and in this case also strongly temporally. And of course, it's questionable to what degree this kind of temporal variation would contribute to evolutionary responses by plants. But the main message of this slide is that pollinator communities vary in space. If you look at that in a more kind of theoretical framework, you can compare two regions with each other. Just call them region A and B. This is taken from one of the special issue articles from the Annals of Botany, to which I'll refer to in a minute in a bit more detail. And if these regions are characterized by different pollinator communities, for instance, short-billed versus long-billed sunbirds, and if the plant species extends its range beyond that of one or two pollinators, it's likely that selection for divergence in floral traits which match the beak length of these sunbirds would occur. And that's precisely what we found in the species of Erica, where the colonization of the area where the short-billed sunbird no longer occurred coincided with the evolution of long floral tubes. And in the literature, we refer to these kind of processes as the evolution of pollination ecotypes. This is another quite striking example where these long-term fly species are distributed unevenly throughout the landscape, and each species is characterized by variation in tongue length. And if we look at the distribution of these three long-tube plant species, we see that all three of them have convergently responded to selection by the tongues of these pollinators, adapting to particular length of the tongues, and thereby forming various pollination ecotypes. This observation that these ecotypes exist was really what inspired us being Steve Johnson, Rob Pico, and myself to kind of invite contribution from all over the world that have studied pollination ecotypes. And in the special issue of Annals of Botany, to which Fernando yesterday referred already, we've amalgamated a large number of studies on pollination ecotypes, including one, for instance, by José Maria Gomez on a more generalist pollination ecotypes, but also from all other continents. And we now come to realize that the existence of these pollination ecotypes is really quite common. And if these ecotypes evolve, and if these ecotypes carry through to speciation events, or perhaps even causing speciation, although that's not something that I want to address in this particular talk, the result is that we see these extreme floral radiations, like the one in Gladiolus, with which many of you are probably familiar with. Having put down this foundation for a theoretical model of pollinator-driven speciation, for which there's a lot of empirical evidence, does not, of course, mean that all floral divergence is necessarily pollinator-driven. This is quite an extreme example of floral divergence, particularly in flower color, which in this case, in this annual daisy, was shown to be heritable, so it's not not phenotypic plasticity, and yet all these different forms of this annual daisy are pollinated by the same pollinator. So it's unlikely that this floral divergence was driven by pollinators. And likewise, there are several studies out that suggest that not pollinators, but herbivores also play an important role in driving floral divergence. So this study by Kessler et al. In Science used transgenic mutants of Nicotiana, and when they knocked out particular genes producing certain scent compounds, these are the knockouts, the incidence of herbivory and ovule predation went up dramatically, affecting plant fitness. So, in this particular case, the evolution of floral scent might have been driven not by pollinators, but by herbivory defense. And a similar example applies from this daisy here, which is polymorphic for the presence of this ring. And this ring was shown not only to enhance pollinator attraction, but also attraction of monkey beetles, which predate on the ovules. So there might be antagonistic selection in plants, not only driven by pollinators, but also by herbivores for particular floral divergent traits. And finally, the same applies to floral shape. So this study on impatiens from a Chinese group showed that the length of the spur and the spur curvature was under selection both by pollinators but also by nectar robbers. And in fact, they suggested that the selection by nectar robbers was stronger than that by pollinators. So basically, in contrast to the first part of this talk, where I suggested that floral divergence there's strong evidence that it can be pollinator-driven, there's also strong evidence for non-pollinator-driven processes. And if you put that in a kind of a very simple diagram, you could argue that floral divergence can be determined by several selective forces. Going back to my first slide, where I showed this extreme diversity of flowers and started out with the question, how or why did this extreme diversity evolve? We would like to get an idea about the relative importance of pollinators versus other drivers of floral evolution. And studies on single plant species are extremely useful, but cannot really answer this question about the relative importance of different drivers of floral evolution. And what we really need for that is comparative studies where we can somehow quantify the contribution of pollinators to floral evolution versus that of other potential selective agents. For instance, in what was done in gladiolus. So in a recent review, Steve and I looked at species-level phylogenies for which pollinator data were available to estimate the number of lineage-splitting events that were associated with pollinator shifts, kind of inferring that those lineage-splitting events may have been driven by adaptation to different pollinators. And we concluded that about 25% of all lineage-splitting events in this rather biased group of clades with fairly specialized pollination systems often was associated with pollination system shifts. But what we didn't do was looking at how these pollinator shifts affected floral evolution. So an open question there is whether flowers evolve only when there's a pollinator shift and not in 75% of the other lineage splitting events, or whether floral evolution is also frequent if there's not a pollinator shift. And that's, after this rather long-winded introduction, that's the aim of my talk today. So to establish the importance of floral evolution, and particularly focusing on the traits scent, which pollinators often use which flowers use to attract pollinators, and shape, which mediates the mechanical fit between plants and pollinators in the light of pollinator shifts. So the objective to reach this aim is by quantifying the evolution of these traits against whether there has been or has not been a pollinator shift. I'm going to focus particularly on orchids, and of course orchids are an extremely florally diverse group, and several fantastic studies are out there that suggest an important role for pollinators in driving orchid floral diversity. So by choosing orchids, perhaps I'm setting myself up a little bit in favor of assigning an important role to pollinators. But let's see how that works out. So this is just a bunch of species that from the orchid genus Satyrium, which is the subject of my PhD studies and also further studies in South Africa. So my PhD was mainly on the molecular systematics of this group, but I felt a strong need that to really understand the evolution of this group, it was necessary to study the pollination biology, and that's what I did during 5 years of postdocing with Steve Johnson and others in South Africa. Satyrium occurs certainly in South Africa with about 35 species in total. These numbers don't add up to 35, but that's because they don't indicate the number of species in a kind of straightforward way. But it's also quite diverse along the high mountains, along the Rift Valley in East Africa, and it even goes into Asia with about 3 species, and several occur in West Africa as well. I'm going to be treating pollination systems in Satyrium, not at the species level. What I mean with species level is the species level of pollinators, but more at the level of functional groups. And this is partly because we simply lack accurate species level data for pollination systems, but I think there's also some justification for using pollination systems rather than pollinator species. Often within a particular population of a Satyrium species, we see that several members of a particular functional pollinator group visit Satyriums and pollinate successfully. We haven't done detailed analyses of the efficiency of these different individual species, like something which we saw a good talk about yesterday. And of course, those kinds of analyses are certainly important to assess which species perhaps might be more important selective agents than other species. But nevertheless, I'm going to treat pollination systems at the level of functional groups. And another rationale for doing that is that if we see that different populations of the same Satyrium species have different pollinator species, but from the same functional group, we don't observe floral divergence, implying that these different pollinated species from the same functional group are similar selective agents in terms of their preference and morphology. This seems a bit boring, perhaps, but it's 10 years of work that are in here in this slide. I had another slide in here that I took out for the sake of time, but pollination work in Satyrium started in 1922, and in fact it was one of the first studies that was done in the Cape Floristic region on orchid pollination. It's actually quite a fun anecdote, so I'm going to tell it anyway. This man, Sidney Garside, went out to study the pollination of one of the very small Satyriums, and he failed to see any insect visitors. So instead of giving up, he decided to look around, and he found these Drosera species. So he inspected the Droseras for insects which might carry orchid pollinaria. And that way, using that kind of lateral thinking, he found out that this particular species of Satyrium was pollinated by fungus gnats. Then it quieted down for a long time until the next kind of pollination work was done in Satyrium. Steve Johnson did quite a few studies in the 90s, and then we decided that for my postdoc, one of the aims was to document as many different pollination or as many pollination systems as possible. And now we had a score of 39 texts out of 98 in total, covering about 40% of the genus. And of those 39, the vast majority is characterized by highly specialized pollination systems. In only 4 cases there's a bimodal pollination system. The diversity of pollination systems in Satyrium is extreme. If you consider what are considered extreme examples of pollination system variations, like Equilegia, there are 3 different pollination systems, or or gladiolus are perhaps 7 or 8. And in Satyrium, we've already got 12 specialized pollination systems, which is really extreme in the light of other studies. The majority of Satyriums turn out to be moth-pollinated. This photograph was added only last Sunday. It's quite crazy. I've just come back from South Africa last Monday. Tuesday I landed in Holland, and Wednesday morning I jumped on the plane to Spain. But I'm really pleased that I can use this photograph for this talk. Several species are bee-pollinated, and then bird pollination is fairly common. And then there's this pollination system that is almost unique to South Africa, pollination by long-term flies, which 4 Satyrium species seem to have. And then there are several smaller pollination systems with fewer representatives. If you look at the distribution of what we know about Satyrium from a geographical perspective, a very scary, biased picture emerges. So, of species that occur in South Africa, we know the pollination system for the majority of them. For those that occur outside South Africa, we know almost nothing. And this is something that I really think should be remedied. But of course, logistically speaking, it's very difficult to spend weeks in the field in remote places like Malawi or Tanzania, where many of these Satyriums occur. If you look at the evolution of pollination systems in a phylogenetic context, you see that a very colorful picture emerges. These are not just known pollination systems that I've optimized on this mean credibility chloroplast tree here, but also inferred pollination systems using multivariate techniques. But the main message of the slide is really that the evolution of pollination systems has been remnant in the genus. So instead of a situation where moth pollination is monophyletic and bee pollination is monophyletic, etc., you see that pollinator shifts have been extremely frequent in Satyrium. This of course creates a very good template to study the evolution of floral traits in the light of the presence of pollinator shifts or on branches where there has been no pollinator shifts. And the first trait that I want to show the results of is that of floral scent, and this is work that I've done with Andreas Jurgens and Steve Johnson, both at the University of KwaZulu-Natal. This is just a sketch of the techniques that we use to capture floral scent. So when you get to the field, you find a plant, the plant is there, you put a bag around it and build this construction such that your scent sampling doesn't crush the plant. And this is what it looks like. So you put a bag, an oven bag that is fairly unscented around the plant, and you capture the headspace of floral volatiles, and you pump air through a tiny hole through the headspace onto this trap, this cartridge with 10x carbon trap. And of course you take a control sample of the environment with an empty oven bag to control for volatiles that are present in the air. And then you analyze these traps using gas chromatography, mass spectrometry techniques. You can store these traps for months, which is really useful if you're doing field work in faraway places. And you get these kind of diagrams out of the GCMS where each of these peaks represents a particular floral compound with its own retention time and what I've not shown here with its own mass spectrum. And those two elements together can be used to identify these compounds. So for the 35 species for which we managed to sample scent, we found 288 compounds. A little bit to our surprise, many of these compounds are actually unique to single species. So about almost 210 compounds are unique to a particular Satyrium species, and only very few compounds are actually shared between species. We found one surprising result, but when we compare this to other studies that have been done kind of at the level of genera, we found a similar result, for instance in these oil collecting bees where you find a similar, orchids pollinated by oil collecting bees, where you find a similar tailoring in frequency of compounds. For those of you who are interested in floral volatiles, I've just done a top 5 of several ways of looking at frequencies or commonness of floral scent. So the most common compound in terms of the way they were found in species is Procrustes, which was found in 19 species. Linalool, which is a very common compound across angiosperms, was found in 16 species. Linalool was also the most common compound in terms of the relative amount in a blend. So, on average, linalool is the most common compound in floral blends of the Satyrium species. And in terms of absolute emission, linalool was also in the top one. The top species. An interesting compound is methylnicotinate, which is fairly unknown in angiosperms in general, but which is obviously fairly common in these Satyriums. Most Satyrium species have very simple scent blends with very few compounds and very low emission rates. So these are the number of compounds in the blend. You see some species have very complex blends, but most species have quite simple blends. And likewise, most species emit scent at very low emission rates, and few species have very high emission rates. This is the number of compounds optimized, or plotted rather, onto the phylogeny, and you see that there's great variation in the number of compounds, and that it's not really phylogenetic instruction. So it's not that particular clades produce many compounds, other clades produce very few, although there is some phylogenetic structure in this clade. For instance, most species produce very few compounds. And the same applies to the emission rates. So you see this rather chaotic pattern with closely related species having very different emission rates. I just want to zoom in on one particular clade where shifts from moth pollination in blue, I'm sorry, you can't really see that very well, I suppose, to bird pollination in black have occurred repeatedly. And if you look at what happens with the number of scent compounds that species produce when they shift to bird pollination, is that here you've got a sister species relationship, moth, many compounds, birds, nothing. Moths, many compounds, birds, nothing. And that happens repeatedly 4 times. And not only the number of compounds, but also the emission rates drop dramatically when species adapt to bird pollination. And of course, that's not really surprising, because we often find that bird pollinated species are fairly unscented. But it hasn't been shown yet in phylogenetic context like this. The most common way of analyzing floral scent data is by multivariate techniques. And this is multidimensional scaling diagram, non-metric multidimensional scaling diagram of Procrustes similarities of floral scent. And each dot here represents a species. And what happens sometimes is, well, it's not really an artifact, but when you have extreme variation between species, is that you get these clouds of species that are actually quite diverse, but that are not diverse in a relative sense compared to species that produce no scent at all, like these. Or Satyrium bucculinum, which has a rich blend that does not overlap at all with any other Satyrium species. Just zooming in here on the cloud, and I've colored the dots according to their pollination system, and I've given them the symbols according to clade memberships. And you see that all the square dots are members of the Satyrium clade, for instance. Round dots are members of the macrophyllum clade, and you see some clustering happening there. And if you analyze that in a statistical context, you can see that species that are phylogenetically closely related are also similar in scent. The same happens though, the same statistical association is found when you analyze groups according to their pollination syndrome. So we find evidence for both phylogenetic clustering, but also for kind of scent syndromes. So species that are blue are moth-pollinated, those that are pink are bee-pollinated, for instance, and those species seem to cluster together in multidimensional scent space. For the remainder of the scent analysis, I'm going to focus on the moth-pollinated species, which are the most interesting ones. When we just do a multivariate analysis of the moth-pollinated Satyriums, we see that some interesting clusters occur. And these clusters are a signature of the dominant compounds that are produced by these species. So, in the middle we have a cluster of species that produces methylnicotinate. This is the unknown compound, or unfamiliar compound, which I mentioned before. And if you look at that in evolutionary context, you see that the evolution of producing methylnicotinate has evolved multiple times independently in moth-pollinated Satyriums. So one could argue convergently. And the same applies to linalool. For instance, the evolution of linalool occurred independently, or at least linalool as a dominant compound occurred independently about 5 times in moth-pollinated satyriums. And the same for this particular compound, it evolved in 2 clades separately in satyriums that are moth-pollinated. So there's strong evidence, and this is actually in contrast to other findings, that scent syndromes can evolve convergently, and that, in other words, that pollinators with a particular preference may have been selective agents for adaptation through producing particular scent compounds. What's quite interesting, though, and perhaps a bit surprising, is that if you classify these different strategies according to their main or dominant compounds, and if you look at those strategies in an evolutionary context, you see that even among closely related species, like in this clade here, different strategies to attract moth-pollinators have arisen. And this is actually quite an extreme example where moth-pollinated satyriums that are dominated by methylnicotinate and linalool are sister taxa. So even though there has been no pollinator shift, there has been a shift in scent production. And we assume that both these compounds are important for attracting moths. But it's obviously a case where the evolution of scent was not driven directly, perhaps, by attracting pollinators, but perhaps by another mechanism, for instance, allowing coexistence of these different moth-pollinated satyriums. Besides compounds which we assume to be involved in the attraction of pollinators, we also observed some compounds which are shared between close relatives, but even those which have different pollination systems. And these I refer to as phylogenetic compounds. So, for instance, tetramurine here is shared in this clade in species that are long-term fly-pollinated and bee-pollinated. And likewise, this compound here, which I won't try and pronounce, is shared between species that are pollinated by oil bees and small flies. Radically different pollination systems, but perhaps phylogenetic compounds that might serve another function. So just to wrap up the scent analyses, which I'll do according to the blend of one particular satyrium species, because I found it quite an illustrative way of classifying different kinds of compounds. Satyrium pygmaeum is moth-pollinated, and these are all the compounds that are found in its blend. And we can essentially distinguish between different kinds of compounds. Those which are unique to Satyrium pygmaeum, in this case there's actually only one that was not found in any other Satyrium species. And then those that it shares with close relatives, even those with different pollination systems. Then there's a number of compounds that Satyrium pygmaeum shares with, not with close relatives, but with other species which are pollinated by moths. And those perhaps can be considered the result of convergent pollinator-driven evolution. Interestingly, not all moth-pollinated satyriums produce these compounds. And then the last class of compounds is perhaps what could be considered pre-adaptation. So, for instance, the compound linalool, which is very common throughout satyrium species, moth and non-moth-pollinated, perhaps may have allowed Satyrium pygmaeum to adapt to moth-pollination. So just to conclude this part of the analysis, perhaps the strongest result is that bird pollination is associated with the loss of scent compounds, and even if there are a few scent compounds left, these are emitted at a very low rate. It appears as if some scent compounds are shared among closely related species with different pollinators, perhaps phylogenetic compounds, one could call them, while other scent compounds appear the result of convergent evolution between species with the same pollination system. And finally, and also quite interestingly, some scent compounds evolve without pollinator shifts, and could either perhaps reflect hyperspecialization or perhaps are mechanisms to facilitate coexistence. So in the last part of my talk, I want to focus on the evolution of floral shape. So the evolution of floral scent obviously could be interpreted as important different means to attract different pollinators. The evolution of floral shape is important for mediating the fit between plants and pollinators. And on this photo, you can hopefully appreciate that fit is actually quite critical. So this long-pronged fly has to maneuver its long proboscis into the nectar spur here. And by doing that, touching the Physidia, which are situated here, and picking up pollinaria, and then depositing these pollen onto the stigma. And this process obviously requires a very precise match between the morphology of the pollinator and the plant. Another example of that is in this carrion fly pollinated orchid, where the fly walks into the flower, and when it does that, these sticky Physidia, which you can see here, stick onto the back of the fly. When it withdraws from the flower, it takes the pollinaria out of the anthosac, and with the pollinaria that are already on the back of the fly, it touches the stigma. And if you look at this cross-section of the flower, you can see that these flowers need to adapt to the size of a particular pollinator to be able to deposit these sticky Physidia onto its back. So it's likely that there's strong selection of pollinators for particular floral shapes. How do we measure floral shape and analyze it? This is something that used to be done by simply measuring particular parts of flowers and doing statistical analysis on these measurements. But obviously, if you're interested in very subtle shape variations, for instance, between these different ecotypes in Satyrium, that look very similar, that only differ very subtly, it's actually very difficult to measure the right parts of the flower precise enough to understand how these flowers differ in shape. So geometric morphometrics is a technique that can be used to analyze variation in shape, and it has a couple of very nice properties. So the first one is that with geometric morphometric analyses, you can really analyze variation in shape while you exclude size from the equation. Secondly, thank you. Patterns of shape variation are retained throughout the analyses, and this is especially attractive for visualizing results. And finally, the way to perform geometric morphometric analysis is through landmark analysis. So you can put a number of landmarks onto homologous points in the flower, and then you can analyze these landmarks. This is a very simple way of suggesting how one analyzes landmarks in a 2D context. Once you have your landmarks, you need to control for variation in the position of the specimen, so you sense them. Then you scale them. This is the step where you remove size from the equation, and you rotate them such that the distances between these points, according to a Procrustes analysis, are minimized. And when you do that, you can come to some very profound insights about variation of floral shape. And for instance, the work by José María Gómez has been a successful, long-standing research program using geometric morphometrics on a species of Brassicaceae. And the same or similar approaches have been used in orchids. And in both cases, 2D geometric morphometrics was sufficient to understand the variation of floral shape. If you look at these Satyrium flowers, you can appreciate that 2D geometric morphometrics of these kinds of flowers would perhaps not be the best representation to understand variation of floral shape. So we designed a method where we can get a 3D reconstruction of these flowers using micro CT scanning, and these are the kinds of reconstructions that you get from these scans. And if you compare a photograph of a real flower with a scan, you see luckily a great similarity, and these 3D reconstructions can then be used to set landmarks. We developed this method, 3D GM in flowers, and we published it using a simple PCA analysis to just show what can be done. But what I want to do in this talk, and I hope you allow me to maybe take about 10 minutes to explain that, thank you, is to look at how these data really can answer questions about the role of pollinators in floral shape evolution. So what are the expectations if pollinators would have been important selective agents on floral shape? The first one, of course, is that species with the same pollinator would be similar in floral shape. And in a kind of ideal PCA environment, this would look something like this, where dots with the same colors would have the same pollinators and would sort of cluster together very neatly. The real world looks a bit more complicated, and I shouldn't be surprised after John's talk yesterday that the real world does that to us. But nevertheless, we do see, even visually, you can probably appreciate that with the kind of pink dots here and the red dots here, etc., that there's significant similarity between plant species that have pollinated similarity in floral shape, that is. But we haven't taken evolution into account here. So what are the expectations from an evolutionary perspective if we compare different floral shapes? Here you see the two same multivariate analyses, but now we're going to superimpose two different phylogenetic patterns onto these. So the first scenario, most of the shape evolution happens among the pink dots, and pink and blue representing two different pollination systems. So most floral shape evolution would be within the environment of the same pollinator, which means that pollinators probably have not been an important driver of floral shape diversification. Contrasting scenarios where there have been huge jumps between species with different pollination systems in terms of floral shape, closely related species. And in that case, we can infer that pollinators may have been important drivers of the evolution of floral shape. So it's really trying to contrast these two scenarios to get the answer. So, in the context of my more complex multivariate analysis, this is the first scenario where there have been radiations in shape within pollination systems, so these would all be each other's closest relatives, and pollinators would not be important drivers of floral shape, versus this scenario where species with different pollination systems that have different shapes are each other's closest relatives. So this is the phylogenetic tree superimposed onto the morphospace. It's called a phylomorphospace. And there are several examples here where closely related species have the same pollinator, color being referring to the pollinator here. But there are also quite extreme examples of closely related species with different pollinators and very different floral shapes. So for instance, we discovered bird pollination in Satyrium rodentum a few weeks ago, and it's sister to Satyrium lumicauda, which is pollinated by moths. And this shift in pollination system from moth to bird is associated with a radical change in floral shape. And we see this actually occurring in several instances of shifts from moth to bird pollination, that these shifts are associated with big shifts in floral shape. To get to a numerical answer to the question of the importance of pollinators, I decided to try and reconstruct floral shape onto the phylogeny, which is not something that has been done often in the literature, at least not in the same way as I've done it. So please bear with me while I quickly go through the steps that are required. So here the color of the branches represent pollinator shifts, so there's been a shift from black to green or different pollinator here. So the first step is to use the Procrustes landmarks and to optimize these onto ancestral nodes. Once you know the ancestral shape, you can actually calculate the distance in floral shape between ancestors and descendants. And if you know the branch lengths, you can use this distance to calculate the rate of morphological evolution. And then finally, you can compare this rate of morphological evolution between branches with versus those without pollinator shifts. When you do that in the case of Satyrium, this pattern emerges. So on branches with no pollinator shift, the rate of morphological evolution or shape evolution is significantly lower than the branches with pollinator shifts. That's kind of expected. We expect that pollinators would accelerate the rate of morphological evolution. But when you then compare the total amount of morphological evolution that has occurred throughout the evolutionary history of Satyrium, you see that there's actually much more morphological evolution happening on branches without pollinator shifts than on those with pollinator shifts. So the conclusion here is that the evolution of floral shape is significantly accelerated during pollinator shifts, but most of the shape evolution actually occurs without pollinator shifts. And this, of course, is a very important conclusion in relation to the question that I first asked. So to wrap up the part about floral shape, geometric morphometrics appears to be a very powerful tool, but I probably don't have to convince you of that, for analyzing floral shape variation. And in the case of Satyrium, pollinators seem to have accelerated the rate of evolution of floral shape, but most evolution of floral shape has occurred without pollinators. So just the last slide of my talk, a general conclusion and outlook. I think I've demonstrated that even in orchids, not all floral evolution appears to be pollinator-driven. And in a way, of course, that is not at all surprising. Although orchids are often used as the prime example for pollinator-driven diversification. These macroevolutionary studies, of which I've shown you some examples, provide patterns, but what they fail to do is provide causal relationships between particular evolutionary processes. So although I've shown that the evolution of bird pollination is associated with changes in floral scent and changes in floral shape, we don't know why these changes happen. So why do bird-pollinated flowers lose scent? Is it because scent repels birds, or is it because if you retain the floral scent, you will still attract moths, which are inefficient pollinators? These questions really need experiments. And finally, again, I don't think I need to convince you as an audience, but I think it's very important that these kinds of comparative studies that combine floral traits, data on floral traits and pollination systems are really important and require a lot more funding, which is often very difficult to find because a lot of this work is simply sitting next to plants for hours and waiting until a particular insect or bird arrives. And this is often not seen by funding agencies as something that would give profound answers, although I think that these kinds of evolutionary analyses are very important to provide answers to why angiosperms are so diverse. Thanks very much.
Bueno, me toca hoy, mañana a mí, moderar la sesión y empezamos por Marcus.
Wonderful talk. I have a comment with a question for you about the scent. I think you have very nicely shown that there is a lot of convergence in floral shape, in floral color, and in floral scent regarding evolution of pollination and so on. We know a lot about, or we have model species for studying floral shape. We have Satyrium, we know all the genes around. The same for color, we know quite well how the metabolisms develop different colors, but maybe we lack model species to study evolution or genetic basis for scent or growing. So what's the situation there? Should we push scientific community to develop a model system to study the metabolism for scent in order to learn more about this evolution, understand why some compounds are genetically preserved and not others?
I think there would be a lot of merit in developing a model system, and I think in terms of the evolution of floral scent, some work has been started in orchids, especially in sexually receptive orchids, where they're trying to identify the genes that are responsible for making small changes to these pheromones that these orchids use to attract pollinators. Orchids are notoriously difficult to use as model systems because it's very difficult to grow them and to wait for them until they flower, etc. I think that there are other means to understand the functional role of floral scent in quite a sometimes simple way. So in a study that was done by Shuttleworth and Johnson, they simply added, and it sounds almost too simple to be true, but they simply added a vial of sulfur compounds to the stem of the plant, and by doing that, they could generate a pollinator shift. So these wasp-pollinated plants suddenly started attracting carrion flies, and not only were the carrion flies attracted to the vial, but they actually went straight to the flower and started pollinating it. So I think that using genetics can be a very powerful way of studying the evolution of floral scent, but I think luckily there are alternatives for plant species for which it's very difficult to grow them.
very nice speech. I have a couple of questions. One is related with the grand steady model. You pointed at the very start of your speech. You say that you need two assumptions, and the second one is that pollinator has to vary distribution, right? And then I wonder whether if you were in a state of plant ecologist, if you were an entomologist, and you see the picture from the other side.
Right.
And then probably the insect is evolving, tracking the distribution of the plant, or?
I see where you're getting it. So you're saying that instead of plants tracking the distribution of insects, it might also be that insects track the distribution of plants.
Probably it's half-half.
I think so. I think there are cases where plant-pollinator interactions are very, very extremely precise in the sense that there's a one-to-one relationship between a particular plant species and a particular pollinator species. In the South African flora, we often see that one pollinator species, I'm thinking about long-term flies in this context, rely on about 20 or 30 plant species. So these pollinators are not dependent on the existence of one particular plant species, but as long as they can reach the nectar in a particular species, they can use that species as a food source. So in that kind of context where you have these very asymmetrical relationships between plants and pollinators, I think there is scope for pollinators to be the main dish to which plants evolve. But in other cases where they're very, very symmetrical relationships, I think it's more, like you say, more a little bit of both. If you see what I mean.
Then probably it's necessary to include that symmetry or asymmetry in the model.
Right.
And the other question is a comment about your study on the evolution of floral shape. It's really very, very interesting. And I'm working also in something similar, trying to get the shape of the ancestor in several species of Brassicas. One thing I'm doing is resampling the individual you have used in your analysis for getting the shape, just to have a statistical measure of the error of the ancestor. You can maybe use that for your sample. If you resample, you can get the shape of the ancestor having a confidence interval, for example.
Right.
You can contrast statistically between different species, the ancestor and the carbon.
I completely agree. There's a lot of this analysis that I haven't discussed in this talk, and one can start thinking about phylogenetic uncertainty, which wasn't incorporated, for instance, which is a big shortcoming. One can think about the fact that there are error bars on the branch lengths, which would affect this analysis. One can think of variation in flowers among populations, or even on an inflorescence, which I haven't even started touching. So this analysis is essentially very crude, and one shouldn't perhaps put too much faith in the conclusion that pollinators have not been important, but I think if you find particular signals, despite all these potential errors, then it's likely that those signals are meaningful. So for instance, the strong convergence in shape of bird-pollinated satyriums is probably something that is biologically meaningful, that can be drawn from this analysis. But I completely agree that.
Probably you can also explore the integration.
Right.
Of the floral shape.
Yes. Yeah.
Could be done.
Yes.
Very good.
Porque hay otra oportunidad.
Okay, Timo, very nice talk. Very inspiring and fascinating. The 12 pollinator systems in one genus. So it must be one of the highest in angiosperm. So I have a couple of questions. The Phylogenetic you are basing all your reconstruction zones is based on plastic markers only. So what about if you put in nuclear?
I was afraid this question would come. In Satyrium, we deal, unfortunately, with some incongruence between the chloroplast and the nuclear gene trees. And again, like in my response to Jose Maria's question, is that one should really incorporate that kind of differences between gene trees. Again, I would say that at the larger scale, where there's no incongruence between these trees, for instance in the several independent origins of bird pollination, etc., we can probably use these kinds of analyses to come to answers. But when it comes to to more detailed analyses of the direction of pollinator shifts, then it becomes problematic to just use one gene tree, of course. So I'm aiming to use both these gene trees and ideally actually get more than just 2 gene trees.
One more thing, sure. Can I? So, if I understood properly your studies, pollinators are important in shaping different florals and then association, involved in association. But there are more factors involved in association of this genus. And in gladiolus, I remember we reconstructed the Phylogenetic, and we found there were a stronger geographical signal than pollinator-related signal for association in gladiolus. Do you think geographic here, I haven't seen you have data about that, do you think geographic here is also very important to shape evolution in the genus?
I think that the question of what has shaped the variation in flowers is perhaps different from the one that then is different from the question what is driven speciation. And I haven't touched on that on purpose because I think it's quite a tricky debate. But I would be happy to defend the conclusion that pollinators have been important for driving floral divergence in Satyrium, whether they've driven speciation in Satyrium or whether that was rather due to, for instance, geographical factors or perhaps adaptation to different habitats, etc. That's something that I would very happily debate still. Is that what you mean?
Yes, but speciation, I mean, the taxonomists define species based on morphology, so it's really related to the shapes you are working on.
Yes.
Of course, there are more characters from seeds or from different parts of the plant, but morphology, flower morphology is basic for taxonomists to tell apart different species. So,
yes.
Yeah, but you're right, I mean, there are more components.
Okay. One more question, Candy?
Hi, Timo. Thank you very much for your talk. Very interesting. And I have another question about the role of antagonists in the evolution of floral shape and biology. In the case of the genus Satyrium, I don't know if you check also if the floral sense and the changes in this implies also a change in floral herbivores. Apart from pollinators, perhaps this implies also that another floral visitors are changing.
Yes.
And it's important just to separate these two processes, no? Because perhaps they are correlated.
Absolutely. I think that's one of the shortcomings of these kind of macroevolutionary analyses is that you identify these correlational patterns. But it's very difficult to get to the cause of why shifts happened. And I really think that experimental approaches are necessary to understand the role of herbivores or antagonistic selective agents with pollinators. And that's something that I haven't done in this genus yet.
Okay, it was very nice opportunity to visit to you.